All right. Good morning, everybody. Welcome to Management of Infections and Inflammations in CF. We are your chairs, Kristen Hudak and Irina Maralda, and we're super excited about this session. When we were putting it together, we really started talking about what management of infections and inflammations really means to us, and we thought that it really requires fine-tuning these four things to get the most adequate management of infection, which is therapeutic interventions, having the right lung microenvironment, having an appropriate host immune response, and then the interplay of microbial communities can really affect the management of infections. And so with that in mind, we came up with these three learning objectives, which were to identify the factors influencing infection persistence, describe how viral infections and mucus environment can alter bacterial virulence, and impact disease progression, and to differentiate the advantages and challenges of novel therapies. And so with that, we have an incredible lineup of speakers that are going to touch each of these areas that I just talked about. And it is my great pleasure to introduce Ana Galdino from Cedars-Sinai Medical Center to come up first. And she'll be talking about leveraging bacterial iron uptake to potentiate antibiotic efficiency.
Thank you. Good morning, everyone. My name is Anna. I'm a project scientist at Peter Jorf Lab at Cedars-Sinai Medical Center. And today I'm going to tell you about how can we modulate the expression of iron transporters in the bacterial membrane to potentiate cefiderocol activity. As we know, Pseudomonas is a major cause of chronic infection in people with CF. And according to the last patient registry, Pseudomonas accounts for 23% of the chronic infection in people with CF. And even post the CFTR correctors era, Pseudomonas, several studies have been shown that Pseudomonas can persist and survive even after 3.5 years post ETI initiation. So these finds indicate that Pseudomonas management is still a central part of the CF care, and the development of new antibiotics to treat those chronic infections are urgently needed. And one of the most recent additions to the antibiotics arsenal was cefiderocol. And this antibiotic is called a Trojan horse antibiotic because it carries this iron binding group that chelates iron from the environment, and once it's bound to iron, it's transported through those membranes iron transporters. And in Pseudomonas aeruginosa, this uptake is mediated by a few proteins, PUA, PUD, PRA, and FPTA. And once the cefiderocol is uptaken and it's in the periplasmatic space, cefiderocol is dissociated and it acts like any other beta-lactam, inhibiting the cell wall synthesis. And although cefiderocol had great activity against gram-negative bacteria, cefiderocol resistance has been observed clinically. And in the beginning of my postdoc, I've been interested in understanding how Pseudomonas evolve resistance against cefiderocol, and we have found that cefiderocol-resistant lineages tend to overexpress pyoverdin, which is siderophore produced by Pseudomonas. And siderophores are strategies that the bacteria evolve to overcome the iron scarcity that the bacteria face when it's in the host. And basically, the bacteria secrete those siderophores, which has great affinity to iron. Then they can sequester the iron, and it's utilized by the bacteria since iron is essential for their bacterial replication and physiology. And Pseudomonas produce two siderophores, pyoverdin and pyokilin. So because we saw this overproduction of pyoverdin and the cefiderocol resistance lineages, we were interested to understand what is the effect of these two siderophores on cefiderocol susceptibility. And we have found a very interesting data where the two siderophores had opposite effects, while pyovidin protects Pseudomonas against cephidrococcal. Pyokilling can actually sensitize the bacteria. So you need less drug to to kill the bacteria, basically. So we already described how pyovidin is protecting the bacteria against cephidrococcal, and now we were interested in understand how pyokilling was sensitizing Pseudomonas against cephidrococcal. So pyokilling is internalized, is uptaken by the bacteria through the protein FPTA. And we know that when we have a mutant which overexpresses FPTA, the MIC of the cephidrococcal is decreased. So we start thinking that this pyokilling sensitization could be mediated by FPTA expression. So to test this hypothesis, we compare the growth of the bacteria culture in the presence of sublethal concentrations of cephidrococcal with and without pyokilling. So while in the wild type strain, the combination of cephidrococcal and pyokilling can completely kill the bacteria. When we have a mutant which lacks the expression of FPTA, this sensitization effect is reverted. So it indicates that FPTA does have a role on the sensitization, and the FPTA upregulation may represent a potential mechanism of increased cephidrococcal efficacy. However, despite this great activity, it doesn't sound like a good idea of giving a virulence factor to a patient. So pyokilling promotes Pseudomonas growth. And in a previous work from our lab, we saw that pyokilling itself can induce lipid peroxidation in macrophages. So we start thinking which other molecule could have this similar effect of upregulating FPTA and enhancing cephidrococcal killing. So we came across with flavonoids, which are compounds that are found in plants in general, and they have like different activities against many different diseases. And it was interesting because some of those flavonoids has these iron binding properties like cephidrococcal and pyokilling. So we start wondering whether those molecules could have an effect on cephidrococcal activity. So we initially test these four flavonoids, luteolin, maurin, 7,8-dehydroxyflavonoid, and myristicin, which both of them share the structure similarity with cephidrococcal. And supporting our hypothesis, previous work have shown that those flavonoids actually can increase the expression of some iron regulation genes in ecology. So to test this, we perform a checkerboard assay where we combine different concentrations of cephidrococcal and different concentrations of flavonoids and follow the bacterial growth after 24 hours. And with that, we have plotted this 3D plot, which each of the squares represent a well in the plate. And when we have a higher peak, it means that we have a strong synergistic effect. However, when we have a deep valley, it indicates that we have an antagonism. After that, we do an analysis which considered the whole plate, and it came up with a synergy score. When it's higher than 10, it means we have synergy. When it's lower than -10, we have antagonism. So we had a similar pattern as we saw for the Pseudomonas siderophores. So some of the flavonoids actually protect the bacteria against cephidrococcal. So myristicin and the 7,8-DHF can protect the bacteria against cephidrococcal. However, luteolin and murin can sensitize the bacteria against this drug. So the next question we had is whether those flavonoids could potentiate other species. And it was interesting because we saw that only Pseudomonas and Burkholderia cynocepacia are sensitized by luteolin and murin. And we came across with the fact that Burkholderia cynocepacia also produce pyokilling and also express FPTA. So we start wondering whether this flavonoid cefiderocol potentiation, like in pyokilling, also depend on FPTA expression. So for that, we generate a reporter string which measure the expression of FPTA. So here we have the curve of the FPTA expression overnight. So in the presence of, when the bacteria is untreated, growing only in iron-depleted media, we have intense expression of FPTA. However, when we add iron to the media, the expression is inhibited. When we treat the bacteria either only with cefiderocol or murin alone, we have no significant difference for cefiderocol, but an increase in the expression of FPTA when we have murin alone. However, when we combine both murin and cefiderocol, we have a significant increase in FPTA expression, indicating that yes, FPTA has a role on this potentiation. So next, we wonder if those flavonoids also affect the expression of the other iron transporters involved in cefiderocol uptake. And we saw that for murin can increase the expression of both FPTA, PUA, and PUA, while luteolin can only induce the expression of FPTA and PUA. And most interestingly, the antagonizing flavonoids cannot affect the expression of those iron transporters involved in cefiderocol uptake. So finally, we decide testing whether this combination would work in vivo. So we infect the mice, and right after the infection, we treat them intravenously, either with cefiderocol alone or with the combination of cefiderocol and murin, and follow the bacterial burden after 6 hours post-infection. And what we saw is that the combination of murin and cefiderocol could actually decrease the bacterial burden in 1 log-fold. So taking together, we could conclude that all of those small molecules like murin that enhance the expression of iron transporters involved in cefiderocol uptake may serve as an effective adjuvant to potentiate cefiderocol. But one problem that we normally face when we are dealing with flavonoids is because those molecules tend to have low bioavailability. So we perform a pharmacokinetic assay to investigate how available murin was. So we give either cefiderocol or murin and collect at different time points the plasma and quantify the concentration of those molecules by mass spectrometry. And what we saw, as we expect, that murin can decrease its concentration in 90% within 2 hours, which may limit the use of this molecule as a adjuvant in a real therapy. So for the future directions, we're gonna use this idea of increasing the expression of iron transporters to perform a high throughput screening against a library of over 100,000 compounds, which we already know they are safe, bioavailable in the host, to identify molecules that increase the expression of those iron transporters and enhance the cefiderocol killing. And with that, I would like to thank my lab mates and our collaborators from USC. And I would like to invite you to stop by my poster if you want to discuss more about it. And on Saturday morning, I'll be presenting another talk where we're going to discuss how the CFTR modulators can affect both the microbiome and the phagome in people with CF. Thank you.
While people are coming up to the mic, I'll take my advantage here and ask you a question. I'm just wondering about the expression of both of these siderophores and if you've tested what environment or what triggers the bacteria to produce either or if they're both being produced all the time.
That's a good question. I didn't test which cues, environmental cues, select for expression of one siderophore over the other. But something that I can tell you is that other groups have shown that pyoridin tend to be overexpressed in the initial infection. And as the infections become chronic, they tend to decrease the expression of pyoridin and increase the expression of other iron acquisition systems, including pyokilling.
Sure. Yeah, sure.
How quickly the bacteria can evolve resistance to the combined treatment?
No, not yet.
Will you repeat the question in the mic?
Oh, yeah. Sen asked how quickly the bacteria evolve resistance to the combination of these two molecules. I didn't test it yet, but yeah, it's something that I have in my to-do list.
Can I ask, I think flavonoids are plant-based
Yes.
products, right? So do we know anything about, say, vegetarians or people's diet and how that affects the antibiotic or any of the expression of iron transport?
It was something that we were actually planning to do initially before we realized that those molecules are quickly metabolized and,
Even
Like,
taken
even-
in orally, they're also
yes, it's even faster, actually. So we came up with this idea, thinking only on the structure of those molecules, but we realized that they are quickly metabolized and which may limit its use. So we gave up on this idea of testing how oral intake of those flavonoids would be beneficial for treating Pseudomonas chronic infection. Yeah.
You have a question for
Hi. I was wondering, have you tested whether or not the cells overproduce pyoridin in response to treatment with the flavonoids? Like, do they respond like they're in a low iron environment?
Are you asking if I saw an increase of pyoridin production when we combined it? No.
No, you didn't.
I tested.
Yeah.
No difference in pyoridin.
You may have already answered this question. How does the bioavailability of
Moraine?
the luteolin compare to fluoride?
So we decide prioritizing Maureen because in our first pilot, Maureen did not behave well in our experiment. So it was hard to solubilize. And even though like in our in vitro experiments, Maureen was always more potent than luteolin. So I didn't perform a pharmacokinetics assay with luteolin just because we decide prioritize Maureen, especially because Maureen also can potentiate several iron transporters, while luteolin can only potentiate two. So we prioritize one over the other. Yes.
Great.
Thank you.
Thank you.
Thank you.
So
So our next speaker is from Dartmouth, Dr. Yasmin Hilliam, and she's going to talk about respiratory viral infection associations with increased Pseudomonas in abundance and CF airways.
Okay, good morning. Just that up. So my name is Yasmin Hilliam. I'm in the Bomberger lab at Dartmouth College, and today I'm going to tell you about some work we've been doing looking at how respiratory viral infection is associated with increased Pseudomonas abundance in the cystic fibrosis airways. So there are no relationships to disclose related to this work. So historically, chronic Pseudomonas aeruginosa has been a real driver of morbidity and mortality in people with cystic fibrosis, and we often sort of historically saw this pattern of initial infection, treatment with antibiotics, the treatment appeared to work and the infection resolved, and then the infection comes back again, we treat, it resolves, and eventually this becomes a chronic infection that is unable to be cleared with antibiotics. And then now, in the era of highly effective modulator therapies, we see that there's this initial massive decrease in Pseudomonas in the airway, but that it lingers around and it's persistent and remains in the airways. And so what we really wanted to get at with the research we're doing is what drives these intermittent increases in bacterial burden and eventually leads to chronic infection. So secondary bacterial infection following viral infection has been, a pretty well-established concept in respiratory viral infection. And so this has been studied sort of previously, looking at how viral infections drive this transition from intermittent Pseudomonas to chronic. And it's been shown that this initial transition to chronic infection occurs more frequently in winter months, so associated with the times when people are getting colds, rhinovirus, coronavirus, things like that. And in pediatric patients, we see that 83% of new Pseudomonas infections actually occur after a preceding viral infection. And so this led our lab to really start focusing on what is the relationship between this chronic Pseudomonas infection and viral infections. And so this is the model we've kind of built over the years. I'm going to sort of show it to you step by step. So initially we looked at the response of epithelial cells to viral infection. We see that there's obviously a really strong interferon response and that when you stimulate epithelial cells with interferon, there's an increase in Pseudomonas aeruginosa biofilm grown on those epithelial cells. We also see that this interferon response drives metabolic reprogramming of the epithelial cells, which also leads to an altered nutritional environment, specifically through increases in lactate by the epithelial cells, and stimulation by lactate also increases Pseudomonas biofilm growth on epithelial cells. We've also seen in clinical isolates previously that there's an increase in transferrin availability, so ion bioavailability, and we've seen that this negatively correlates with the alpha diversity of in these clinical samples, and that it also correlates with the competitiveness of Pseudomonas against other gram-negative bacteria grown in the clinical aspirates that we measured the ion in. And so all of this led us to want to ask, do respiratory viral infections change the microbiome of the cystic fibrosis airway? And so to look at this, we conducted a observational longitudinal study at the University of Pittsburgh Medical Center. This ran from 2017 through to 2021. In the particular study I'm going to talk about today, we had 36 subjects, and they were sampled every 3 to 6 months from the right maxillary sinus. Importantly, these were sampled endoscopically, so this is not nasal sampling, this is directly inside the sinus, and then from expectorated sputum. And the data we got from this was 16S microbiome data, a viral panel, and then from the sinuses, cytokine data. So just to run through the viral detection that we had in these samples, the viral positivity rate was slightly higher in our sinus samples than in sputum, not surprising given that most of these are upper respiratory infections, and human rhinovirus was our most frequently detected virus. So the first question we wanted to answer is the bacterial community composition in the airway altered during viral infection? And the way we wanted to look at this initially was through diversity metrics. So I'm going to show you a little bit of imaginary data for a second, just to explain this concept that I'm going to use. So I'm going to be talking about distance to centroid. This is a beta diversity measure, and it measures the variation of a microbiome composition. So on this plot, this fake plot that I'm showing you, each of these red points represents the microbiome composition of an individual sample. And I'm going to be using data generated from Morrissey to Horn distances. This is a dissimilarity metric. So the samples that are more similar to each other cluster together, the ones that are not cluster apart from each other. And so the analysis that I did was to generate a centroid. So this is essentially an average of the microbiome composition for all of the samples from each patient that were pre-viral infection. Then we can measure the distance of all of their other samples, so during virus and post-virus, to that pre-virus centroid. And so essentially the measure that we're getting here is how different is the microbiome during each of these stages from the pre-virus microbiome. So, as I just said, greater distance to centroid, greater difference from pre-virus community composition. And so what we saw was really interesting. So on the y-axis here, I'm showing you that distance to centroid. And then on the x-axis, I've got the samples binned by their pre, active, and post-viral status. We see a significant increase in the distance to centroid during viral infection. So that's shown in yellow here. And what's most interesting to us is that we don't see a restoration of the microbiome composition post-virus. So there's still a significant difference in distance to centroid between our pre and post-virus conditions. Interestingly, though, we also looked at alpha diversity, and there are no changes to alpha diversity during viral infection. So Shannon diversity is generally really low in our samples. Maximum is kind of 2. So really indicating that these samples are very undiverse. We also looked at Fisher diversity, which takes into account much more of the rare taxa. And so that's also low. And what we can really gather from this is that these samples are kind of dominated by very few taxa. So the next question for us is which community members are driving these changes to the microbiome? And as I said, we know from our diversity metrics that these are very, undiverse samples. So we figured the best way to look at this is to identify core taxa. These are taxa that are really important across the whole sample set. And so the way we identified these was by gathering any taxa that had a mean relative abundance across all samples greater than 1%, and a prevalence of greater than 75% in all samples, shown by the dotted lines here. And so you can see we found 3 taxa that met these cutoffs, and I think to no one's surprise, these are Staphylococcus, Pseudomonas, and Streptococcus. And in the plot that I've got on the right here, this is just mean relative abundance of these 3 core taxa across all the participants in our study for all of their samples. And so you can see that Pseudomonas and Staphylococcus are really dominant across these samples, and Streptococcus is really only making up sort of 1 to 20% across most of these people. So now if we look at those core taxa, sort of before and after viral infection, here the viral infection is represented by this vertical black line. So these are samples that were taken on a date when a person had an active viral infection. Anything on the left is pre-viral infection. Anything on the right is post-virus. And we've got sort of scatter plot smoothing overlaid here so that we can see the kind of general trends of relative abundance for these 3 taxa. And what we see is this kind of blip. When we get a viral infection, there's this sort of increase in Pseudomonas and paired decrease in Staphylococcus relative abundance at these positive visit dates. So now if we group those just by whether a sample is viral negative or viral positive, we see that we have a significant increase in Pseudomonas relative abundance during viral infection. And so this plays really nicely with kind of everything I've told you so far. We see that viral infection is causing Pseudomonas to increase in its abundance. We do also see this really interesting bimodal distribution here, especially with the Staphylococcus. We do have a subset of our population who are chronically infected with basically 100% Staphylococcus in their microbiome, and these people don't have any shift regardless of time, viral infection, anything like that. They're just constantly infected with Staphylococcus, and so we're not sure what causes that, but it's certainly something that's interesting and worth looking at. So to give you a quick summary of what I've told you so far, the airway microbiota are significantly perturbed during viral infection. This perturbation is driven by significant increases in Pseudomonas relative abundance and by probably a decrease in Staphylococcus, and this is supported by current literature implicating viral infections in bacterial expansion. And so most importantly for us, the next question is Is there an association between this host response to viral infections and the altered microbiome composition that we see during a viral infection? And so what we did to look at this was a 37-plex unbiased cytokine screen. What I'm only showing you here are the 7 cytokines that had significant associations. And just to orient you a little bit with the data, because I know there's a lot happening here, on the y-axis, we're looking at Pseudomonas relative abundance. The x-axis is normalized cytokine concentration. And I'm currently only showing you the viral negative samples. So there's really not too much of a relationship happening with any of these cytokines and Pseudomonas abundance when there's no viral infection. However, if we now lay the viral infections over the top, we see that there's a significant difference in the relationship, the correlation, this positive correlation, between cytokine concentration and Pseudomonas relative abundance during viral infection in these patients. And we were most interested to see that IL-29, or interferon lambda 1, was significantly associated with Pseudomonas relative abundance, as this really nicely dovetails with the in vitro work that the lab has carried out previously. So to summarize this all into a model, we see that acute viral infections change the airway microbiota composition, and that these generally tend not to return to baseline after the perturbation. Viral infection leads to increased cytokine concentrations in the airway, and that this is then associated with an increase in Pseudomonas. And so we know this is really important because for people who are modulator ineligible, Pseudomonas is still a really big problem in cystic fibrosis. And for people who are modulator eligible, we see that Pseudomonas isn't being eradicated in the airways. And so we may still see this pattern of driving intermittent recurrence of infection and eventually leading to recolonization with chronic infection. Future work leading on from this is more about expanding our understanding of the role of nutritional immunity and immunometabolism on macrophage function in the airways. So how does that sort of altered nutritional environment and change in cytokines impact macrophage efficacy in its killing and their polarization? And we also want to look at how this altered nutritional immunity affects Pseudomonas biofilm growth and its ability to compete with other microbes in the airway. We also think there are some interesting clinical implications for this. This is quite a translational study, folding everything in from in vitro work all the way through to clinical work. And there's some work been done on the ion chelators. So the idea being that if we could stop Pseudomonas from being able to utilize that increased ion during viral infection, we may be able to stop the Pseudomonas expansion that occurs as a result of that. There are also metabolic inhibitors that are currently under clinical trial for cancer treatment that would stop that metabolic reprogramming that would lead to the secretion, the increased cytokines and increased nutritional availability in the airway milieu. So with that, I would like to acknowledge everyone in my lab that helped get this work published, not published, it's about to be published, sorry, not quite yet. Get it up here, get it ready for you for presentation today, including people who left the lab previously, especially Catherine Armbruster and Summer Attia, and everyone at Pittsburgh who helped out whilst we were there, and to all of our funders, and thank you for your attention. I'll take any questions.
So I have a question. Should we be giving some anti-interferon therapy to people in the setting of a virus? Obviously, this is theoretical.
I would say no, because I think it's probably driving a bunch of the really important clinical aspects. I also think, I should clarify that most of our viral positive samples came before the introduction of modulator therapy. We were unfortunately hit with kind of a double whammy with COVID, when basically everyone stayed indoors for 2 years and didn't get any viral infections, and then modulator therapy came along. And so this is much more of a pre-modulator look at things. And so I think the effect of viruses on the CF airway in the era of modulators could be quite different. I think there's probably still likely to be an effect. But I definitely wouldn't like to say that we should be giving people anti-interferon treatments.
Excellent talk. Very clinically relevant. Thank you.
Hi, Liz Bergener from CHLA.
I can hear you, okay.
You can hear me. Okay, great. Great talk. As a peds pulmonologist, very interesting to think about the viral effects and whatnot. But my question was exactly about that, about ETI or about modulators. And did you include the patients on modulators in this analysis? And was there enough to see any differences, or was it just too few samples?
I think so. Out of the people who were on modulators who got viruses, I think it was like 1 or 2. And so unfortunately, there's just no power for us to make any statements about the effect of ETI on this like new era of treatment.
Thank you.
Thanks. Great talk. I'm Claire Wainwright from Brisbane. I wonder if you could comment on the use of antimicrobials, inhaled antimicrobials, and what you were finding. I was kind of interested because with the use of inhaled tobramycin, we do see a reduction in Staph aureus, and I wondered whether during viral infections and increased exacerbations, you might find that people actually used their inhaled antimicrobials, and whether that might impact what you found.
Yeah, so all of these patients were all on kind of completely personalized treatments. So everyone was on a completely different cocktail of antibiotics. And so, again, it's a pretty small cohort study, and we just unfortunately weren't really powered to say anything about the antibiotics. But most of these patients were consistently using antibiotics and using an antibiotic regimen. We did see, I did look at the impact of exacerbations, and there were no differences in whether or not there was just no relationship really with whether or not it was an exacerbation visit or just a regular clinic visit.
Yeah, this is a really nice talk. First question is, do you know if the absolute abundance of Pseudomonas also increases during the viral infection?
Yeah, so a big limitation obviously is that this is 16S. We were not able to get at absolute abundance just because we don't know exactly what species are in there, but we did do total 16S quantification, and there's no significant difference between our viral and non-viral groups for like just 16S total abundance. So I think we can relatively confidently say that this is an actual increase in Pseudomonas and a decrease in other members of the microbiome during this viral infection.
Okay. The other thing I was wondering is, have you considered the possibility that during the viral infection, the patient is coughing more deeply and bringing up more sputum from the lung, and so the dilution factor of the saliva is decreased, and so that could increase the relative abundance of PA coming up during viral flares?
I had not considered that. I think that is something I could definitely look back into. I think we have recorded weights for the sputum, so I think this is something we could look at. But I think it is also important to point out that this relationship was also really strong in the sinus, where there's maybe more mucus coming out in the sinus at that point. I could have a look. I could have a look back through our kind of, Catherine kept very diligent paper records that I can look back through.
Thank you.
Thanks.
Excellent questions. I think we're ready for our next speaker. Thank you.
Thank you so much.
All right, so next up we have Anna Lee, who will be telling us about experimental evolution to weaponize bacteriophages for Burkholderia.
Hi everyone, I am really excited to be here. I do have one conflict of interest to disclose, which is that I'm the CEO and founder of Corion Health. But without further ado, excited to tell you guys about my work. I'm currently an MD PhD student at the University of Pittsburgh and Carnegie Mellon. And before I get into the science, I think it's important to talk a little bit about the context, because just getting to this project has taken me 12 plus years. So this is me and my friend Megan in the pit orchestra clarinet section back in high school. As you might guess, she has cystic fibrosis. I found out when she was hospitalized, and I had to learn both of our parts, which was inconvenient for me and really awful for her. So not wanting to lose my best friend, I kind of panicked and tried to get into cystic fibrosis lab. So my first experience was actually in high school, sweeping the floor and organizing the shelves as I volunteered in Scott Rendell's lab at UNC. Stayed there throughout undergrad, and then around 2018, I saw the paper coming out of Pradeep's lab saying that even if you fix the underlying issue in cystic fibrosis, the infections persist. So I emailed Pradeep and I was like, what gives? And he's like, why don't you come over here and find out? So I ended up taking my 2 gap years in Pradeep's lab, and I am now in Von Cooper's lab in the MD PhD training program, co-advised by Derry Van Tyne, working on custom engineering bacteriophages to actually eradicate these infections. And just a fun aside is when I got into the MD PhD program, Megan had just graduated from law school. She's now a high-powered lawyer, among also doing 6 hours of treatment a day. So no excuse for any of us really to not do our best. But she messaged me, is like, hey, what are you doing? And I'm like graduating from law school in Florida. And I'm like, oh, I'm moving to Pittsburgh. She's like, is it sunny there? I'm like, yeah, so sunny, very sunny in Pittsburgh. So I feel like I kind of tricked her into moving here, but it's nice because she's still there and we get to hang out like all the time. So yeah, that's my personal story. Okay, anyway, back to science. So Burkholderia, as many of you know, is associated with worse prognosis and Cepacia syndrome, which is a rapid sepsis and decline in patients. It's pretty awful. I don't know if I mentioned, but Megan has Burkholderia, which is what brought me to this. It's intrinsically antibiotic resistant, so it does tend to form chronic infections, which actually makes it an attractive candidate for phage therapy, especially because it tends to be of a single lineage in most chronically infected patients. But the problem is that there's very few phages available. So why is this? Well, due to the need for phage host specificity, it is very difficult to find a clinical match. So our solution to this is to engineer better phages. So you can imagine many groups, including our group at the University of Pittsburgh, have phage libraries where you might have an ancestral phage that we know has strong efficacy on some propagation host, which may or may not be clinically relevant. Okay, well then you get a clinical isolate from a patient whose doctor has said, like, hey, they're a good candidate. Let's see if we can find something. We screen them against the entire library, and we only find one match, and it is too weak to really realistically consider deploying it in clinic. So ideally, we have an evolved phage, a junior phage with strong efficacy on this clinical isolate. And while there's many ways to do this, including some of my colleagues at CMU who are working on bottom-up engineering of specific proteins to maybe increase binding, we let natural selection do all the hard work for us by utilizing experimental evolution. So I'm going to walk you through this adventure in 3 parts. The first one is how to train your phage. The second is what makes a good phage. And the third is the bacterial resistance when things go wrong. So without further ado, the critically acclaimed How to Train Your Phage, which one critic has said is a spectacular and a must-see. So before we start training phage, we need to know what we're training toward. So we've defined 3 killing potential indices, also known as KPIs. The first one is phage productivity. So, it doesn't matter if the phage can't actually reproduce. So this measures how quickly and how well it can reproduce. The second one is bacterial killing. We don't care if a phage can reproduce if it can't actually kill bacteria. And the third is heritable genetic changes, so that this is not regulatory. For example, phages that integrate into the bacterial genome would not be as good candidates for this. But we want to make sure that whatever phenotypes they have that we're selecting for will be carried on into clinic. Okay, so I use the CAR method, which is to challenge, assess, and repeat. So we start by challenging the bacteriophage with bacteria at a known multiplicity of infection. For those who aren't virologists, this is just the ratio of phage to bacteria. So we add some phage into this turbid bacterial culture, and then after a day or so, we look at the OD 600, or the density of the phage-treated culture compared to the no-phage control. If it's lower, as in if the phage reduce the bacterial concentration compared to the control, then we'll actually decrease the MOI. So that means we either reduce the phage or increase the number of bacteria in the next passage. If it's higher, we either increase or keep the MOI constant. And you can think of this as going to the gym and lifting weights. You're never going to get stronger if you're only lifting 5-pound weights. You have to, as you get stronger, you want to keep on challenging yourself with more and more things. And so in a similar way, we continue to increase the selective pressure. So then, once we have our plan, we isolate the phage from the bacteria, titer it, sometimes expand it again if we need more, and then we repeat the experiment over and over again. And the goal is to continue doing this until we have a phage so strong that you can drop one phage into a turbid bacterial culture and it nukes the whole thing immediately. So you can imagine our goal is to have fewer phage, kill more bacteria faster, right? So let's look at what happened when we did this. Well, we actually did see that phage productivity increases over time. So here's passage 1. You can see at hour 0 and 24 hours how many phage went into the culture and how much came out. And then we can actually look at this across passages. And you'll see that as we continue to challenge the phage, even though we're putting in a similar amount of phage, they are able to reproduce faster and create more phage at the end, which is really important because when you deploy this clinically, you tend to filter out some phage in the purification process. And by the end, we're actually passaging after like 8 hours or 7 hours instead of 24 because we want to select for the ones that are lysing the bacteria faster. This is a little bit messy to look at in aggregate, so what we've done is reduced it to a rate, so you can see the simple data points, and we also conduct this in 4 independent experimental replicates, so you can see that it's pretty reproducible across these different replicates. Yeah. Okay, the second one we looked at is bacterial killing. So here you can see the optical density of one of our replicates compared to the control, which is in black. And you can see that at first, we knew this phage didn't have great activity here, but as we go on toward passage 11, we see that it's quite consistently able to control the bacterial population, but this isn't the entire story because not only is it able to control the bacterial population, it is controlling more and more bacteria at a time. So you can see here that the MOI is actually decreasing over time. So we wanted to take all this data and kind of simplify it into one metric. So we ended up taking the negative of this and multiplying it by the difference, and you can combine those to get a simple metric for bacterial killing efficacy. And again, you can see the 4 replicates show a similar pattern over time. So basically what we're doing is expanding the phage population, bottlenecking it under selective pressure, and then continuing to expand it so that we can get stronger and stronger phage. This is the same data I've just shown you. If you want, these are the lines so you can kind of see the trends. But what we actually care about here is whether this is heritable, right? This is KPI number 3. So we did sequencing at every couple of passages to see like what's going on at the DNA level. And what's really interesting is you can see around passage 7, I don't know if this pointer's gonna work. Yeah, you can see around passage 7 as the KPIs are actually increasing more significantly, that's when we really see some genetic mutations start to rise toward fixation. So the next thing we wanted to do is isolate clones of phages as opposed to populations so we can actually understand what the direct mechanisms of action are, or rather what makes a good phage. So we took the most common mutant of every experimental replicate, and we did some more experiments with them. So this takes us to part 2. So for a little bit of context, the phage that we're using, BCCO2, is predicted to be of the genus Pedoverinae. We did some electron imaging of it, and yes, we saw that it has a capsid head and a contractile tail sheath. As you might have tracked from the animation, these represent the 4 mutants that we're looking at. Please note it is one phage diagram to make it easier to look at, but these are 4 separate mutants. Okay, so the first thing we looked at was whether the phage can be more productive on the new clinical strain as compared to the original host. This is something called efficiency of plating. Actually quite easy to calculate. You just do titers on the new strain and divide by titers on the old strain. And you see for most of these, it's actually, it has improved at least 2x, but for this tail fiber one, it doesn't look like it has improved, which was a little confusing to us. But note that the way that we're measuring titers here is on solid agar assays, and tail fibers are most implicated in the part of phage binding where it's non-reversible, or sorry, where it is reversible, and so they tend to benefit more from being in liquid culture where they can actually kind of rely on these diffusion and electrostatic forces to actually find the bacteria. So what we did next was look at adsorption. I will also say all this clone-specific data is very, very fresh, like last week fresh, so please bear with me as I'm going through it. Some of it's not fully complete yet. And also wanted to do CF Foundation colors, but if anyone's colorblind, I do have these lines show up every now and then. Hopefully that helps. Okay. So to look at phage adsorption, we plotted the decline of free phage over time when the phage were exposed to an excess of bacteria. So for this, we can just use mass action kinetics and then some statistics, and we'll pull out a adsorption rate constant. Note that the typical adsorption rate constant for phage is something like 10 to the negative 9th. So we've only done this with 2 of the mutants so far, but we're already seeing that with these 2 mutants, it is much higher than typical, which might explain why the tail fiber mutant is still showing these improved KPIs, even if it's not showing an improved efficiency of plating. Okay, moving right along. Of course, we don't care about binding or productivity if they can't actually kill bacteria, as we've mentioned several times. So the next thing we did was look at growth curves in the presence or absence of phage. So here you can see MOIs along the top line right here. If there's no phage added, we have these nice logarithmic bacterial growth curves. But as soon as we add some phage, we see this characteristic peak and then a collapse of the bacterial population. So this next assay is actually inspired by my work in Pradeep's lab when I was working on broth microdilution assays, looking at minimum inhibitory concentration of antibiotics. I think you can think of this as something very similar. We basically increase the phage concentration relative to the same bacterial concentration twofold with each column over. We can then plot or add a line for the limit of detection, which for our plate reader tends to be at around 0.01, and then point out the first MOI or phage concentration where there was no growth above that line. You can then map it to the MOI and plot it, and you'll see that there's quite a range in what the minimum inhibitory MOI is for these phage. But hold that thought, because if we pull efficiency of plating back in, so this is the data I just showed, and then flip it around, we actually see that they correlate pretty well, which I think I was pleasantly surprised by. So just keep in mind the one on the left, minimum inhibitory MOI, is done in liquid culture and is more of a measure of the phage's ability to suppress the bacterial population. We don't actually count phage titer in that experiment, whereas the efficiency of plating, we're actually counting phage titer on a solid agar assay. So it was pretty interesting that the direction of phage strength correlation, so just to orient you again, if you need less phage to control the bacterial population, that's good, they're stronger. And if they produce more on that bacteria, that also means they're stronger. So we saw that the correlation of phage strength is actually correlated between these two technically different assays. Which gives me hope that maybe this might be more predictive of what happens in clinic, since we know that clinically it's probably a mix of both. We can also think of this as lower MIC is correlated to higher efficiency of plating, which kind of makes sense when we think about it from a mechanistic standpoint, but it's still nice to see the actual evidence to confirm it. Okay, so this was going great. I was pretty stoked about these results until I forgot about the plate reader and went home. And then the next day it was still running, and it had gone from 24 hours to 48 hours. And to my horror, many of the bacterial populations had rebounded. So I was like, okay, this is no bueno. What is going on here? So this brings us to part 3, the bacterial resistance. Had some ideas, but I needed some help at this point, so we reached out to our friends at Drew Bridges' lab at Carnegie Mellon University. They specialize in using confocal microscopy to take videos of bacteria growing over time. In their case, mostly for biofilm, but it's still helpful just to look at what's going on. So we worked with them. Here's a bacteria-only one. You can see this video that came from their lab of the bacteria growing over time. Okay, nothing to see here. But as soon as we add phage, you can see this characteristic peak and then collapse of the population. And as I'm about to show the video, I want you to pay attention to what happens right around 20 hours, where it looks like the phage are rebounding. So you can see they look normal at first, but then suddenly around 20 hours, you see it looks pretty different. There's some sort of mass aggregation effect, and I totally thought I did something wrong and this was crazy happening, but if you look at all the results from all the phage, it happens quite consistently. I think it's most prominent with the tail fiber mutant, but it happens a little bit with all of them. So talked to Drew, and apparently this is actually a known phenotype of LPS mutants. So when LPS has different charges, sometimes the bacteria start sticking together, and I was like, yeah, good story. But then we did sequencing, and if you kind of filter out the noise or those that didn't rise above 50% frequency, we actually found that WCA and WAA actually are mutants that have been known to be critical in the LPS pathway. So I was like, okay, Drew, I believe you next time. Candidly, I'm not sure what this transposase one right in the middle is doing here, but we're running out of time, so we must move on. Okay, so then we also wanted to look at the fitness, because, ideally if a bacteria is antibiotic resistant or phage resistant, we gotta have something to work with, right? So maybe it's less fit. So we did growth curves of these phage resistant populations, and we actually did, based on area under the curve from the growth curves, which is a commonly accepted metric of fitness, we actually did find that the not phage resistant bacteria were somewhat more fit than the ones that were phage resistant, but as soon as you add phage, all of them drop in fitness, with the non-resistant ones dropping the most. So not entirely sure what to make of this. I do think it's somewhat helpful to know that maybe clinically, it's not just the phage eradicating the bacterial population. We also have antibiotics to work with. We also have the immune system. So hopefully the phage can knock down the infection enough that another player can take it out completely. And with this, we've come to the conclusions. So I hope that my work has convinced you that there is some good preliminary data and some promise for using experimental evolution to train better bacteriophages. And in the future, we want to optimize this for generalizability across different phage host combinations. So here's the current one we have, but we'd like to show that it works well in a couple others. And once we have a generalized algorithm, we can invest in robotic automation. And since I'm studying machine learning at CMU for my PhD, I must include that we would love to have a predictive algorithm. So the evolution experiment is still, it takes like 2 to 4 weeks, but I think it's still faster than just like randomly screening wastewater phages, especially for patients who don't have a good option. But many patients don't have that much time. So the goal is to have, if we have a new clinical isolate where we need a phage match, we can run it through this predictive algorithm that has been trained on all the phage libraries and potentially immediately output a phage or a somewhat modified phage from the library to give to patients. So with that, thank you very much for listening. I would like to thank everyone in the Cooper Lab, the Van Tine Lab, and the Bridges Lab, as well as my funding sources, the CF Foundation, and the Pollen Disease Soros Fellowships. And please feel free to reach out. I also will have a poster here, which is poster 68, so feel free to roll through and keep chatting if you want. So thank you.
Hey, Anna, that was really good. Jay Dandekar from the University of Washington. LPS mutations often influence susceptibility to antibiotics, and I was wondering if the evolved bacterial strains that you see when you treat them with these phage that emerge have different susceptibilities to, a host of polymyxin or other cell membrane targeting antibiotics?
Yeah, that is a great question. Okay, I did test that, but only with one antibiotic, so we actually didn't see any change. But that does remind me that we need to look at some other antibiotics. If you have any that you know of that tend to be more susceptible or less susceptible after LPS mutants, that would...
Yeah, I'd start with the polymyxins.
Okay, I'll try those. Yeah, I used tobramycin just because from my time in Pradeep's lab, that one was like my favorite. It was so stable, so reliable. Love tobramycin. But yeah, I should probably branch out a bit more. So thanks. Yeah.
What environment did you use to do the evolution experiment in? Was it?
Oh, yes. I used a M9 plus minimal media.
Okay.
This is one that's been pre-formulated to be a simpler version of the synthetic CF sputum media. Originally, we did it in LB, but then we realized that's probably not the most representative of the lung, and we kind of want to train it in an environment as close to the lung as possible.
And have you done any comparisons to see how effective these trained phages are against a broader host range of vocal area? Like, how has focusing on a single strain curtailed its ability to be more effective in other bacteria?
That's a great question. We did do efficiency of plating on a couple other clinical isolates, and we found varied results. Sometimes it was more effective, sometimes it wasn't. I think it would require further analysis to determine why that is. Yeah.
Hi, great talk. My name's Katie Wetzel. I used to be in Graham Hatfull's lab, so I think about this sort of stuff all the time. And I was curious about your tail fiber mutant. Right. It said, was it an L286L?
Yeah, it's a synonymous one. Yeah.
So what do you think about that?
That's a great question. It definitely had me scratching my head at first. So I talked to some other phage experts in the lab, and apparently sometimes there is a mismatch in codons between phage and their hosts, and to the extent that some phage carry their own tRNA. So the working hypothesis we have is perhaps this is a result of some sort of bacterial mismatch. So even though it looks like it is a synonymous mutation, maybe it's not in reality. But again, more assessment would be needed to actually find the answer. But we've seen that happen in other phage, so it's not out of the question.
Okay, great. And I have a follow-up question,
sure.
Which is maybe not as relevant if it's not a coding difference, but I guess, there's some phenotype.
Yeah, it might be something else, yeah.
Right, but my question is, do you know enough about these phage tails to, put things into AlphaFold, get an idea of whether these residues are outward, inward, how it might be contributing?
Yeah, that's a great question. I think AlphaFold right now is not as good, to my knowledge, at predicting how mutations change structure. They can predict a given structure, so I candidly haven't tried that hard at it yet because I wanted to look at the change in mutation. We have run some molecular dynamic simulations, which are a little different, kind of a more old school method, but we didn't see significant differences. But again, I think those parameters could be further fine tuned. So definitely still on our plate.
Yeah. Great, thank you.
Cool, thanks.
Thanks, James. I don't know.
That was a great talk. So I'm just curious, what happens when you mix the 4 phages together or just do pairwise tests? Do you see the same phenotypes?
That's a good question. I haven't done that yet. Yeah, as mentioned, this is all like very fresh data from last week, but yeah, I think it would be really cool to mix all 4 phages together and see what happens. I think it is. It was very surprising to me that they were in such different parts of the phage. I thought that there'd be more parallelism, like they were all tail fiber mutations or something, but yeah, these 4 different populations all showed similar rise in KPI, but without having the exact same mechanism. So it would be really cool to like put them together. Yeah, if you want to share contact info, I'll send you the data fresh off the press. Yeah. Yeah. Okay.
Thank you.
Our next speaker is from the University of Alabama at Birmingham, Dr. Caitlin Sebastian. Well, PhD candidate. Sorry about that. I apologize. I didn't read the candidate word. I'm just trying to double click on this. Okay. She's going to talk about increased concentrations of mucin impede Staph aureus growth in synthetic CF sputum. Okay.
Shaky up here. All right. Thank you for that introduction. And to just start off, I have nothing to disclose. And so we've talked a lot about today about how bacterial infections are still a problem even after highly effective modulator therapies. So kind of along that idea, as early as when ivacaftor came around and people started looking at how is impacting bacterial infections, 1 year post-ivacaftor, they started seeing that Pseudomonas aeruginosa was decreasing in some studies. However, we weren't seeing these same effects with the bacteria Staphylococcus aureus. And so that made us start wondering why this is occurring. And so for just some basic information, Staphylococcus aureus is a common pathobiont colonizer of the human nasal cavity. And when we look at how staph is persisting in the CF lung after ETI treatment, we see that there is an initial decrease after the first month on ETI, but it still persists all the way up until 6 months, indicating that there's something in the environment that's giving Staph aureus the ability to survive. And so we were curious really because MRSA specifically, or methicillin-resistant Staph aureus, is really associated with an increased mortality in CF populations as well as overall worse disease outcomes. And so to really start understanding how staph is persisting in this environment, we started thinking about the mucus layer of the CF airways. And so in a healthy lung, the mucus layer is really meant to act as a physical and chemical barrier to protect you from things like toxins, allergens, and bacterial infections. However, in CF, because of this dysfunction of CFTR, you really get this disruption in the mucus layer where it becomes a lot more compacted, entangled, and becomes more difficult to clear out the airways. And this is particularly due to this increase of secreted mucin, specifically MUC5B and MUC5AC, as well as an increase in ectocellular DNA from various sources. And so due to this, it creates a really niche environment for bacteria to live, as well as making it more difficult to expel the mucus. And so primarily this talk is really going to focus on MUC5B. And so MUC5B is a secreted mucin from the submucosal glands and really abundant in the CF airway itself. And so to kind of get a further idea of what these are starting to look like when they're secreted is they tend to start off as these smaller strand polymers. And then as they move through the airway, they oligomerize and become what we know as the mucin network mesh. It's a lot of words there. However, in the CF airway, we start to see that due to proteases released from the host, such as through immune cells, specifically neutrophils, or from bacterial infections, we start to see that these mucins get degraded and back into their smaller polymers. And some studies have showed that these smaller mucin polymers may have pathogenic effects specifically on the host, but not much is understood on the impacts of bacteria. And so we started looking into the literature of what do we know about Staph aureus and mucin interactions in the airway. And some of the earliest stuff we could find is that shows that Staph aureus can bind to mucin, specifically from the nasal epithelial, and it prefers to bind to mucin-coated wells compared to that of BSA-coated. So this at least lets us know that there is something occurring between staph and mucin. Other groups have noted that while staph cannot degrade mucin itself, it does rely on other bacteria such as anaerobes to degrade mucin, or other bacteria to cleave various side chains such as sialic acid to act as other carbon sources that it can utilize and grow. Additionally, another group has seen that Staph aureus can exhibit this dispersal phenotype when it's grown on agar containing mucin. So there's something occurring between Staph and mucin, but it's just not well understood. And so we do think that there is a way that Staph aureus has adapted to really persist in this complex mucin environment with the various links of mucin strands. And so to really start parsing this out, we utilize the well-characterized synthetic cystic fibrosis sputum media, also known as SCFM. And so this really helps to mimic these CF nutritional environments, specifically with the increase of ions, amino acids, and other aspects. And this will also allow us to really control the additions of extracellular DNA as well as mucin. And so to really start getting the idea of the different strands, we found that there was 2 commercially available bovine submaxillary mucins at different polymer sizes. Specifically bovine just because it is, or submaxillary mucin specifically because it's abundant in MUC5B. And so here we will be using a low molecular weight mucin. Going forward, it'll just be referenced as LMW, and it's around 200 to 500 kilodaltons, representing those smaller polymers. And then we're also using a higher molecular weight mucin, or HMW, where it's about 400 to 4,000 kilodaltons, so it's much larger and a little bit more variety. And so first we really wanted to see how these 2 different size mucins are impacting Staph. And so first we grew Staph aureus in an SCFM with no mucins. And we saw that I was able to grow above inoculum, which is represented that dotted line, indicating that Staph can grow in this media. What was interesting is when we grew it in a low molecular weight mucin environment, we saw a significant decrease in Staph aureus recovery, as well as being way below the inoculum baseline. We also used microscopy to look at how Staph was behaving in biofilm, and we saw that it primarily created these small aggregates, not really creating any type of biofilm itself. Interestingly, when we grew it in the high molecular weight mucin, we saw that it was no different than the no mucin control, and still significantly higher than that at the low molecular weight mucin. And it's able to form a really nice biofilm that we were able to confirm through quantification. And so we really wanted to see how early this negative effect of low molecular weight mucin has on Staph. And so here we also added extracellular DNA to see if that might help to recover it at all. And so first we started at 4 hours, and even as low as 4 hours, we start to see that Staph is significantly decreasing in the mucin-containing conditions compared to that of extracellular DNA. And we continue to see this trend occur even at 8 and 12 hours, with the biggest difference being at 24 hours. And there was really no differences whether or not eDNA was with the mucin or without, indicating that eDNA is not doing much with just overall growth. However, we were wondering if maybe it's changing its biomass in any way to utilize it as like a scavenging. And so here, again, we just grow our GFP Staph in SCFM with no polymers, with eDNA, and there was really no differences between these 2 conditions. And again, when we grew it in a low molecular weight, we again see these aggregates, and we still see this when we add extracellular DNA to the media, indicating that eDNA is not sufficient enough to increase overall Staph aureus biomass. And so next we wanted to see if we lower the amount of low molecular weight mucin might help to recover Staph, especially since highly effective modulator therapies are kind of meant to correct the mucin concentration. And so right now our media is really meant for people with CF around 5 to 6 milligrams per mil, where we wanted to go back down to what is considered a healthy concentration of around 2 milligrams per mil. So again, we grew Staph again in the low molecular weight mucin with 5 milligrams per mil. We're still seeing that same phenotype. And interestingly, when we decrease the amount of low molecular weight mucin, we see this increase of overall biomass and increase of overall aggregate size. So as those small mucin polymer chains start to go away, we start to see an increase of Staph again. And so This also is confirmed through our CFUs, where we see a partial restoration of Staph aureus in the 2 milligrams per mucin compared to that of 5 milligrams, but it's still not the same as what you see in the SCFM alone. And so we've talked a lot about how mucin is impacting staph, but we were also wondering if it goes the other way around. How is Staph aureus impacting mucin? And so to do this, we worked in collaboration with the Burkitt lab at UAB, looking at the viscoelasticity of the mucus. And so in CF, we see this increase in viscoelasticity where it becomes a lot more stretchier and more viscous. And this is what really leads to the more difficulty of clearing out the mucin. And so first we looked at viscosity at both the low and high molecular weight mucin. In the low molecular weight, we don't really see any differences between the unaffected and affected. And this also holds true with the high molecular weight, indicating that staph itself is not impacting viscosity. Next, we looked at elasticity. And at the low molecular weight mucin, we see that there is a significant increase in elasticity when mucin is present. And we see this further increase with the high molecular weight mucin, where it's further increasing the elasticity. And this was interesting to us, especially because in low molecular weight mucin, Staph aureus does not grow as well, but it's still doing something to the mucin. And so finally, we wanted to see if this is more so a broad effect. So we are looking at other Staph aureus strains as well as other respiratory pathogens. So first we're going to focus on other Staph aureus strains. And so to do this, we use GFP fluorescence tagged Staph aureus. This is primarily due to the low molecular weight mucins being really turbid, and it's really hard to just get a normal growth curve. And so here we're able to utilize a measurement of GFP as an approximation of bacterial burden. And so first we grew it in SCFM with no polymers, just to kind of get a baseline of where these bacteria are with their GFP expression. And these are six different laboratory strains that are very common in the Staph aureus field. And so when we added mucin, we actually saw a major drop in GFP expression. We did confirm that this is higher than that of the baseline autofluorescence by quite a bit. And so this at least lets us know that staph was there. It's just not growing as exponentially that we see in SCFM alone. We also did this with other clinical isolates, specifically ones from CF, people with CF. And again, we saw in SCFM alone, all the isolates were able to grow. And once we added mucin to the mix, they all decreased, still higher than baseline, but not doing too great. And so next we wanted to look at other respiratory pathogens, since we know that CF is often including other bacteria. And so this is just a reminder of what was shown very early, where we see the decrease of staph in the low molecular weight mucin. And so first we looked at Pseudomonas aeruginosa in the high and low mucin, also with the addition of eDNA represented by the horizontal stripes. And while there were some differences between conditions, overall Pseudomonas was able to, one, grow above that of inoculum at the dotted line, but also maintain that persistence despite conditions. We also looked at a early CF pathogen, non-typable Haemophilus influenza, and while it did not necessarily grow above that inoculum, it mostly maintained itself. There was still no significant difference between conditions, whether it was low or high. And we also looked at another bacteria specifically that is seen in a mucin environment. This is an oral streptococcus, and interestingly, we saw that there was a decrease of CFU recovery in the low molecular weight mucin, and it has better growth in the high molecular weight mucin. And again, this may be kind of more so gram-positive focus. And so in conclusion, low molecular weight mucin really is impeding Staph aureus growth and biofilm production, and that eDNA cannot restore the growth of this or the biofilm. And that if we reduce the low molecular weight mucin concentrations, it does partially restore Staph aureus biofilm and growth. And this seems to be a very broad impact of Staph aureus strains. And also we're seeing that Staph aureus is able to change the elasticity of this both low molecular and high molecular weight mucin, but not the viscosity. And with that, thank you to my labs, also to the Burkitt lab for helping with the rheology and funding sources. That's not mine.
A very interesting talk. Wondering what you think if the high molecular weight you're seeing increased staph growth. What about if you have the proteases like neutrophil elastase? Does that sort of drop it back down to what you would see with a low molecular?
I would think so, since other groups have kind of showed that like any type of protease that is able to degrade mucin, it does kind of break them up into smaller pieces. So I would think it would possibly reduce it again.
Sounds good. Thank you.
Hi, I was wondering, if I got this right, in some of your essays, you see that over time, the staph don't grow up. And I was wondering, do you think they're dying, or do you think they become non-culturable over time?
So I've gotten that a lot. It's possible that they're dying, especially just when we're looking at the overall biomass itself. It does decrease with it. But that is something we probably need to like further confirm. And how we would go about that, I guess, is a live dead stain is probably going to be the easiest way.
Thank you.
Hi, great job. I wanted to ask about the exact formulation of the SCFM you were using. So we've recently started using the zinc depleted SCFM, just from the original paper that was shown to be like slightly more accurate. And actually for strep sanguinis, I see when I use it that it like tanks its growth. Like it's super, super happy in an anaerobic environment with SCFM2. It loves it. It grows super well. And then as soon as you deplete that zinc out, it just crashes right out like below its inoculum. And so I just wondered if you had A, which formulation you were using, and B, like if you had thought about modulating any of those other kind of like inputs that are in there.
Yeah. So I still make my SCFM from scratch. So I make all the stops.
I feel sorry for you.
Yeah. So I make all the stops and everything. So it is kind of like, it includes everything that you would see in the original recipe, and then doing the DNA and mucin, all that. So ours does have zinc. And the mucin is just kind of seems to be dependent. So we've noticed that a lot of companies that sell the bovine submaxillary mucin, they're not comparable. And so I think that depends a lot, but yeah, I still hand make it, homemade.
Yeah, we just maybe tried like the zinc lowering the amount. It did really like have an effect on some of the gram.
Yeah, I can try that.
Hi, Caitlin. Very nice talk and nice data. So have you any idea or hypothesis why staph is particularly increasing elasticity of the mucus?
Yeah, so one thought I have is that staph is able to make phenosoluble modules, PSMs, is I guess a short way to say it. And those are kind of acting almost like soap, if you want to think of it that way. So through those productions, it could just be making it more of a slippery, and so it kind of adds to that elasticity.
Have you any, or usually elasticity is also like increasing by ROS, so increasing of or oxidation of the mucin crosslinks. So have you tried to investigate that, or does it may also play a role there?
Yeah, so I forgot to mention, but the rheology data showed was with the mucin cross-linked, but all the other data that I showed doesn't have the cross-linking.
That was a great talk. I'm just curious, have you ever done an experiment where you just start growing staph using the high molecular weights mucin and then swap for the low molecular weight?
That is something I've thought of and wanting to do. And so that is something I'm interested in. I have played a little bit around with ratios, and generally if there is enough high molecular weight, it just seems to kind of overpower the small. But I would be curious to kind of like starting growth in the high and then trying it in the low.
It's kind of related to the last question. Yeah. Have you tried co-cultures of Pseudomonas and staph in the low molecular weight?
I have not yet. Not gonna lie, I'm not a big Pseudomonas person. Not a fan, more of a staph. But that is something that would be interesting. My PI is very excited about that comment. Very interesting to try, though.
Thank you. Great questions. Keep them coming. Thank you.
All right, so our final speaker is Matt Biggert, and he will be telling us about AVP10, which is a novel antiviral peptide with reduced activity in CF airways.
Thank you so much for the introduction. Yes, so I'm going to be talking about AVP10, a novel antiviral peptide with reduced activity in CF airways, and I have nothing to disclose for this presentation. So our lab's primarily been interested in RSV, a negative sense RNA virus that primarily infects ciliated epithelial cells. So you can see here work by Ray Pickles and Peoples, who showed using a modified RSV that expresses GFP in infected cells, that it seems to localize to those cells that have that are ciliated. In non-CF patients, RSV is a leading cause of hospitalization in young infants. However, in RSV patients, even going into older ages, RSV tends to lead to persistent inflammation, bacterial co-infection, and long-term decline in lung function. So, in the healthy airway, where CFTR is transporting bicarbonate and chloride and hydrating and buffering the airway surface liquid, antiviral peptides can be released where they can interact with viruses, bind them, aggregate them, and clear them. In the CF airway, where CFTR is either not present or not functional, we end up with a dehydrated, acidified airway surface liquid and a thickened mucus. So when antiviral peptides are released into it, they appear to have impaired viral clearance, leading to these more severe viral infections. And we thought that the airway surface liquid might be home to or host to some interesting antiviral peptides. So the lab went about obtaining sputum from non-CF and CF donors. We then separated the native peptides from the large proteins using molecular weight spin columns, and without digesting the native peptides, we analyzed them using mass spectrometry. This gave us a peptide library, which we then synthesized using solid phase peptide synthesis and screened for biological activity, looking at the effects the peptides had on cell growth, whether the peptides were toxic, and if they showed any antiviral activity. So, looking at this chromatogram of the non-CF and CF sputum, when being run through the mass spectrometer, we saw a clear change in the relative abundance of native peptides in the CF sample. And this translated into non-CF patients having a relatively low number of peptides per subject at about 100, and CF sputum having a relatively high number of peptides at almost 1,000. And this becomes really clear when you look at principal component analysis to analyze the difference in their peptidomes. The non-CF sputum, or peptidome even, shows a remarkable amount of similarity, whereas the CF peptidome, while there is some similarity, they are much more heterogeneous than the non-CF, and there are some significant outliers, but in either scenario, all of the CF peptidomes were significantly different from the non-CF. And we synthesized all of the native peptides and 1,000 of the CF peptides, or 1,000 of the most common CF peptides. And one family that stood out to us was that there were multiple peptides derived from PRH1, which is proline-rich allele protein 3. And so this little graphic up here shows the relative positions of the peptides we found from PRH1. We exposed these to cells and then infected them with a modified RSV that expresses GFP when infected. What these representative images show, and the data on the right shows, is that when uninfected, there's no GFP expression. The RSV showed high GFP expression. AVP10, which is a name we gave because it was an antiviral peptide with an isoelectric point of 10, was able to significantly reduce GFP expression, and all of the remaining peptides represented by this one here, or characterized by this one here, did not significantly affect RSV infections and did not affect GFP expression in cells. So we wanted to see if AVP10 would retain this antiviral activity when inside of its large parent protein, PRH1. So in the infection, we saw high levels of GFP expression, which was significantly reduced by AVP10. But PRH1 itself did not show any antiviral activity. So what we learn from this is that AVP10 is encrypted within the PRH1 protein and needs to be released to carry out its antiviral activity. We also found that AVP10's antiviral activity is dose-dependent in cell lines. So here we have uninfected cells with no RSV, with no GFP expression, RSV-infected cells with a high GFP expression. And as we increase the concentration of AVP10, we see a decrease in GFP expression in a dose-dependent manner. And we found it had a IC50 of about 3.28 micromolar. So we wanted to start looking into how this antiviral activity was being carried out. And to do that, we labeled some purified virus with a fluorescent tag and very simply let them settle in a cell-free environment. And what we saw with RSV was relatively dispersed viral settling, whereas when we added AVP10 to RSV, we saw these much larger aggregates forming. So when we looked at the viral area, RSV had a relatively low viral area. When AVP10 was added and the aggregation, and they formed aggregates with RSV, we saw a significantly higher viral area forming, or fluorescent area forming. And when we scrambled the sequence of AVP10, the sequence of the peptide, we lost this activity, which indicates that AVP10's aggregated activity is dependent on its sequence. But we also wanted to make sure that what we'd seen wasn't just an artifact of fluorescently labeling the virus. So we used transmission electron microscopy, and here you can see free virus in the RSV-only control, but when treated with AVP10, we start seeing these clusters forming as the virus starts to aggregate. And in a separate experiment, we were actually able to visualize this with the naked eye. So when we took some concentrated RSV and added AVP10, we could physically start to see aggregates forming and that the remaining liquid was beginning to clear. So we took these viruses, separated them into aggregates and supernatants, and saw that the majority of the virus was contained within the aggregate. In the supernatant, we had almost abolished RSV's infective potential, but the aggregate still was less infective than the RSV control. So we wanted to understand whether this aggregation, or how this aggregation was providing an antiviral effect. So we wanted to see if it reduced the capacity of the virus to bind to the cell membrane. And we did this quite simply by fluorescently tagging RSV, adding it to the peptide, and exposing it to cells at 4 degrees for 2.5 hours. This allowed the virus to bind to the cell surface but not be internalized. And after 2.5 hours, we washed away the unbound virus, fixed the cells, and read the fluorescent intensity. And what we saw was that RSV was binding to the cell surface, but that AVP10's addition was decreasing the membrane-bound viral fluorescence, indicating that it had reduced the capacity of, or that aggregation was reducing the capacity of the virus to bind to the cell surface. And we carried this one step further, and instead of fixing the cells, we returned them to 37 degrees, and 1 hour later looked at intracellular fluorescence, so whether the aggregates were able to get into the cell. And what we found was RSV showed a relatively high intracellular fluorescence, so lots of virus getting into cells. That AVP10 significantly reduced the amount of virus that was getting into cells, and when we scrambled the sequence again, we lost this activity. So having the beginnings of an idea of a mechanism, we started to look in mice. We exposed them to AVP10 and then infected them, and 72 hours later sacrificed them. And what we saw at the 72-hour mark was that RSV was making the mice lose a lot of weight very rapidly. They lost almost 5 grams in 72 hours. But when we treated them with AVP10, we were able to reduce this weight loss and provide protection to the mouse from viral weight loss. We then started to look at the H&E staining of the mouse lungs. So in the uninfected, you can see there's plenty of free airway space for efficient gas exchange. When the mice are then infected with RSV, inflammation starts, immune cells infiltrate, and the amount of free air space collapses. But when we treat them with AVP10, we see somewhat of a dose-dependent response where free air space is beginning to be restored to the airway. So uninfected cells have a lovely high free airway area. RSV collapses that, and treatment with AVP10 is able to significantly raise it and begin to restore the mouse to a more uninfected state. So a lot of the work I've been talking about, you might have noticed we were adding AVP10 first, so we were testing its prophylactic potential. We also wanted to look at whether it had therapeutic potential. So here we exposed cells either to AVP10 1 hour before being infected with RSV, or we exposed cells to AVP10 24 hours after infecting HBECs, human bronchial epithelial cells, to test whether it was therapeutically able to reduce cell infections at 72 hours. And what we saw was a significant right shift in the dose response, with prophylactic performing better than therapeutic. But the therapeutic use of AVP10 still was able to clear the infection at the higher concentrations. Which brings us back towards cystic fibrosis. So when we took non-CF and CF HBECs and tested AVP10's potential, at 3 micromolar we found that non-CF HBECs, there was a significant reduction in the number of cells expressing GFP that were infected by RSV. So AVP10 was carrying out its antiviral activity. But at the same concentration in CF HBECs, we saw a much higher rate of infection and no significant difference between CF cells treated with and without AVP10, which indicates that CF has inhibited some of AVP10's antiviral activity. So what conclusions can we take away from this? Well, in normal cells, the free virus will infect ciliated epithelial cells and spread, and this is more severe in CF patients. When we treat the virus with AVP10, it appears to aggregate it. Now, the exact mechanism of aggregation is still something we're trying to determine. Whether it is interacting with one or multiple viruses as well as itself, or whether it is interfering with the viral membrane and disrupting it, causing them to fuse into larger aggregates, is not known. But in either case, these aggregates are less able to bind and infect cells, making them easier to clear by either the immune system or mucociliary clearance in the lungs. And in the CF airway, this activity appears to be reduced. And there's a whole range of possibilities for why this might happen. It could be that AVP10 is being degraded by the higher protease levels of the CF lung. It could be that some of the proteins that are overexpressed are binding and activating AVP10. Or it could be that AVP10 is pH sensitive and is simply no longer able to carry out this activity in the CF airway. But the net result is the same, that the CF airways are more susceptible to these infections and are infected at a higher rate despite AVP10's presence. So what we really want to do moving forward for the next steps is to determine the mechanism of action of this aggregation and to begin looking at what is changing in the CF lung that is inhibiting AVP10's activity, so we can better understand what its role in the innate immune system of the airway is and whether it has any therapeutic potential moving forward. So I would love to thank my mentor, Rob Tarrant, and the entire Rob Tarrant lab. None of the work that has been shown here could have been done without a huge amount of work from a range of different people. I'd also like to thank the wider KUMC community who've been a huge help with a range of different aspects of work and providing material support, as well as various people who've provided material support, and obviously our sponsors and funders in the NIH and Cystic Fibrosis Foundation. Thank you so much.
I have a question. Do you know if the presence of AVP10, if it varies among patients, and is there any correlation between expression and RSV?
Not entirely. So we have, I don't know entirely. We know AVP10 is expressed in some previous RNA-seq data we use, so it's expressed in the airway epithelium. I believe it's primarily expressed by club cells. I don't know what effect RSV specifically has on it. I don't know what effect CF might have on its expression. There's a few potential hints in other systems where it's present, but I don't know for certain.
Okay, great.
Hi, really nice job. Joshua Chandler from Emory. I wanted to ask you, both with respect to how you screened for peptides originally, and then looking at what you think about what might happen to AVP10 in CF, how do you factor in post-translational modifications that might still be present on the peptides? Was that something that you were able to capture in the original screen, and then also in the biosynthesis? So I'm thinking about things like methionine sulfoxidation or chlorination, or all these different things that could happen that might be relevant both in the healthy context and then maybe in CF.
You're absolutely right that the modifications that may be occurring, you would be better off discussing it with Rob Tarrant. I personally didn't do quite a lot of the screening. I would say from the data I've seen, I don't believe we particularly looked for post-translational modifications, and when we synthetically synthesized them, we didn't include, it would be extremely difficult to include that number of different modifications. The majority of the peptides are under, I'd say, 9 to 14 residues long. I think the hope is that the activity would primarily be carried out in the relatively disordered peptides by the sequence itself rather than by the post-translational modifications. But yeah, I don't know for that aspect. We certainly found a surprisingly large number that were active. So it seems these peptides do have an important role to play. And in the future, and certainly in some of the modifications we've done when trying to develop some of these peptides to a more therapeutic role, we have started to introduce modifications to them to make them more active. So you're very possibly right that we might be missing some of the data with it, but it would be very hard to do thousands of peptides.
Understood.
Including all of those modifications.
Thank you.
No problem.
Thanks. Really interesting talk. I had two questions. One, have you guys for the sputum looked at the microbial composition? Are there any correlations between certain microbial community members and AVP10 expression? And then second, a more RSV specific question. We know RSV is in filaments when it actually is propagated in a ciliated epithelial cell. And so your aggregation phenotype, I saw HEP2 data. So if you propagate in HEP2 versus a primary bronchoepithelial cell, do you see differences in the aggregation? Because the morphology of RSV is probably different.
No, we haven't actually looked for that yet, but that would be really interesting to do. We primarily work with the virus straight off of the HEP2 to be concentrated up and then
it's hard enough
applied to them. We haven't looked at that, and we probably should.
Really interesting talk. Sam Durfee from Emory University. I was wondering if you've tested the efficacy of AVP10 against any other viruses?
We're literally in the process of doing that as we speak. It's based off of its aggregated activity. We actually have some really nice, simple ways of testing whether it is aggregating other viruses. We have been collaborating with another lab at NC State who have kindly been exposing other viruses to AVP10, and they're just in the process of getting that data back to us.
Okay, cool. I guess based maybe on what you find there, thinking about phage particles, I was wondering if phage could be an absorption, like essentially stealing the AVP10 from like its normal function in CF.
Yeah.
I want to thank all the excellent speakers and thank you for your engaging questions.